My name is Torben Hansen. I'm group leader here at the Center for Basic Metabolic Research. And it is my pleasure to introduce the next speaker, Stefano Romeo from the Karolinska Institutet. Stefano has done fascinating work in the intersection between human genetics, metabolism, and liver disease, focusing on the genetic architecture of steatotic liver disease and its cardiometabolic consequences. Stefano has been part of many interesting discoveries, including finding the genetic variants in PNPLA3, which has learned us a lot of steatotic liver disease. And his research aims to translate genetic discoveries into improved risk stratification and targeted intervention for metabolic and liver-related outcomes. So we look very much forward to your talk, identification of two types of MASLD with different clinical trajectories. So
thank you very much. Thank you, everyone, and the organizers for having me here. It's my pleasure and delight. So I will talk today about this liver disease, and these are my conflict of interests. And so, as you know, steatotic liver disease, or MASLD, is worldwide a big problem. I mean, roughly 1 in 3 have the disease. We are lucky in Europe that it's 1 in 4, so we are a bit behind. And this goes together with the obesity epidemics. But if you take someone with type 2 diabetes or obesity, it's almost 2 in 3 with steatotic liver disease. And if they have them both, it's almost inevitable that they will have the accumulation of fat in the liver. So this is what happens. This is just a normal liver. This is the normal liver architectures in people at the histology. You see these huge empty vacuoles. These are very large lipid droplets full of triglycerides, neutral lipids. And this is when you have liver steatosis, more than 5% of the content. This is much more than that. It must be at least 30%. What happens is also the hepatocytes become larger and pale, sort of like a balloon, and this is what is called ballooning. It's a sign of hepatocyte damage. But the liver also becomes infiltrated with macrophages, and so you develop also inflammation. What is really important is the production of fibrosis after long-standing damage to the liver. You get liver fibrosis. And from an organ perspective, many years of production of collagen gets due to cirrhosis, and this is still a condition that can only be cured by liver transplant. Another bad outcome for the person with the disease is the hepatocellular carcinoma. This is one of the most aggressive, and still the prognosis once you have it, if it's not at the very initial stage, is very poor. I mean, we're talking about months. So this is a more than 20 years old paper from Hannele Ikinarvin in Helsinki. She was the first one showing that the more fat you have in the liver, the more fasting insulin, indicating that there is an association between liver steatosis and increased insulin resistance. This is very similar if you look at liver fat and circulating triglycerides. And liver steatosis, MASLD, is also a risk factor for diabetes. This is the adjusted model in this study. And this is true whether you look at men, but also in women. This is after adjusting for all the other risk factors for diabetes. But steatotic liver disease is also an independent predictor of coronary artery disease and atherosclerosis. And this is a case control study from France, in which you see that the presence of steatosis predicted the thickness of the carotid artery. And also it predicted the amount of calcium in your coronary arteries. These are like classical atherosclerosis signs. And they were also doing better than diabetes, for example. This is a study from the Swedish registry from Hannes Axström Karolinska, and he just stratified the population in those with MASLD and control. You see that in this very long follow-up, more than I think it was more than 18 years average, those with MASLD have almost a 4-fold increased risk in developing cardiovascular disease. But we're talking not only about coronary atherosclerosis, but also heart failure. And this study shows an independent association between MASLD and heart failure. And finally, there's also epidemiological evidence that the disease predicts the presence of kidney failure. This is whether you have diabetes or you don't. So it's an independent predictor of chronic kidney failure. And so the question is this really MASLD causing a multi-organ disease? And the answer is we don't know, because this is only epidemiology. I could certainly think that because when you have MASLD, you have an atherogenic dyslipidemia, high triglycerides, and more LDL, then you would have a coronary artery disease. But when it comes, for example, to chronic kidney disease, you have also diabetes, and diabetes is a well-known risk factor for that. You also have hypertension in the conundrum. That's also a risk factor for that. For diabetes, you have a lot of visceral adiposity. So it's hard to tease out causality from epidemiology. Another question is, what do these people with MASLD die from? So these are the cause of death related to cancer and cirrhosis. And you see that if you look at those without MASLD, these are pretty much non-existent. So there is a large excess of that when you have MASLD. However, quantitatively, you have more cardiovascular disease than liver-related events. So a question is, can we identify those that will only develop liver disease from those that actually will get cardiovascular disease or both conditions? So I will start by talking today about 3 common genetic variants causing steatotic liver disease and the PNPLA3 TM6SF2, also some data in the MARC1 area. This is not exhaustive of all the ones that have been discovered. What I just wanted to point out is that most of these variants act on liver steatosis directly, and then they drag you all the way down to the very end of the disease on cirrhosis and cancer. Just want to spend a few words on the acquired mutations. So the one not in the germline, so the one that you get after you're born. And I find very interesting that acquired mutation for cirrhosis are on genes related to triglycerides and insulin resistance, FOXO1, lipid droplets, side B. And these are actually protective, trying to protect against cirrhosis. Instead, when you look at hepatocellular carcinoma, they are fundamentally different. These are all genes involved in cell cycling, so these are one causing it, indicating that although these 2 conditions are very close in time, seems to have very different pathophysiology. So I mentioned that most of the genes are related to liver fat. And how is this liver fat regulated? So when you have an excess of calories, triglycerides get synthesized in the ER, and then they can get packed with apolipoprotein B, ApoB, forming VLDL, very low density lipoproteins, and then they get secreted. And the liver is the only organ that can't secrete triglycerides in the form of VLDL. If you were looking at lipid profile, when you look at triglyceride levels, those are a function of the VLDL. It's just the yellow part here. However, when you have positive energy balance, triglyceride can also be stored in lipid droplets. When you have negative energy balance, fatty acid can be released from lipid droplets and be oxidized in mitochondria to generate energy. So whatever changes the balance between release from lipid droplets, beta oxidation, or VLDL secretion, it would affect lipid content and the outcome. I will start by talking, as it was alluded to, PNPLA3. So I was in Helen Hobbs in Dallas at UT Southwestern as a PhD student and got the chance to look at the data in the Dallas study. And here you see a Manhattan plot, and what you can clearly see, even without knowing how to interpret the data, that there's only one point here. And this was the only SNP that was associated in significantly, without any bias for multiple testing, with liver fat content. So you see here that carrier of the variants have higher liver fat in a dose-dependent way. So PNPLA3 is a protein that sits on the lipid droplets. And what it does, it allows the free fatty acids to be mobilized from the lipid droplets, so to get out. So the frequency of the variant in homozygosity in Europeans is 5%, those that are healthy. But if you look across the liver steatosis, then inflammation, cirrhosis, and HCC, you see that there is a tremendous enrichment, making so that from 1 in 20, you get almost 1 in 2 that are homozygotes when you have liver cancer, more than 1 in 3 when you have cirrhosis. And this gives you very large odds ratio for the disease. You see it goes from almost 10 here in these studies, 4, 3, and these are odds ratio that are very large for a common variant. I mean, this is very unique in the genetic architecture of common disease. If you look at diabetes, obesity, we're talking about 1.2, 1.3 odds ratio. So it's a very special event or situation, condition for the disease. So what is interesting is that the effect of the variant is not the same between men and women. So this is a Nature Medicine paper, published with Luca Valenti, been working a long time with him. And what we showed was that it doesn't matter if you look at the liver fat or this ballooning or inflammation or NASH, fibrosis, significant fibrosis, the effect of the variant is always larger in women. This is the p-value for the interaction that, for being an interaction, p-value is very large. So women with the variant have a larger risk for liver steatosis. And if you want to know more about it, in the paper we show that this is due to a estrogen receptor binding site that is in the promoter region of PNPLA3 that increases the protein production in women. This is interesting because if you look at mice models, the knock-in mouse, if you challenge with fructose diet, you see the effect of the variant only in female mice and not in male mice. So indicating that even in the mouse model, this dimorphism, sex dimorphism is there. I only show you cross-sectional data. These are prospective data, and you see that this is a Japanese cohort, also very long follow-up. These are liver-related events for cirrhosis and cancer. And you see that basically if you do not have the variant, nothing really happens to you. You have like a hazard ratio for carrying the variant of 16 in this cohort that, again, it's very large. So this is, just to show that this is a robust variant increasing the risk. And we thought it was important to studying it. So after about 10 years of modeling and genetics and in vitro studies, we, came up with a model in which we thought that this is the way it would work if we wanted to target this protein to treat MASLD. So PNPLA3, as I said, it allows fatty acids to get out and so makes the lipid droplets smaller. But there's not only PNPLA3, there's also this other protein, it's called PNPLA2 or ATGL, that also does the job. So this is a redundant system. It's not only one protein doing it, it's redundant. And then there is also this cofactor that is called CGF58 that is important for the hydrolytic action in triglycerides in both proteins. So the idea was PNPLA3 is not working, so it's a loss of function. However, it is also a gain of function in the sense that it sequesters more of the CGF58 cofactor. So it also doesn't allow the other protein to get it. And the idea was that if you would to remove the mutant protein, you could restore the system, allowing the other protein to work. And so we tested this in knock-in mouse model. This was a collaboration with AstraZeneca. And we fed the model with a NASH-inducing diet and then treated the mice with an antisense oligonucleotide. And you see that the effect of the ASO, it was well tolerated, at least in body weight, no difference. These were the treatment was started. Also no changes in caloric intake. It did work very well on reduces the protein at an mRNA level. This is the mutant mouse and this is the wild type mouse. And you see also the protein level, the protein was almost gone. This is lipid droplets purification. This is the only way to see it. You need to concentrate it by extracting only lipid droplets for the liver. The fascinating thing was that the model was correct, and we saw that in the mutant mouse, there was a reduction in steatosis, in liver inflammation, and liver fibrosis. While in the wild-type mouse model, the effect was blunted and only present for steatosis. And so I'm not involved in this paper. So this is a proof of concept that the model works in people. This is our 2 phase 1 studies in which you see a dose-response reduction in liver fat content after targeting the mutant protein. These are homozygotes for the PNPLA3 protein with an siRNA GalNAc conjugated that goes specifically in the liver. So the jury is out there, but I think this is proof of concept that potentially we could treat, instead of having the medicine, you give the same medicine to everyone, fatty liver, everyone take the same, you could do genotyping, finding those mutants for the PNPLA3 and target directly the PNPLA3 protein. Still early days, time will tell. So I wanted to tell you a little bit on TM6SF2 now. And TM6SF2 was discovered by Helen Hobbs also with another genome-wide association study. I was not there at that time. And a study subsequent in mice showed that TM6SF2 sits in the endoplasmic reticulum, and the variant is a loss of function. In this mouse model, they could show that what the protein did in mouse was allowing enriching triglycerides in the ApoB. So the yellow part would be larger because of TM6SF2 secreting more fat. And the knock-in mouse had much less of that. However, the number of particles were not changed. So the number of particles are function of apolipoprotein B because there is only one ApoB for each particle. So no changes in the amount of particles, but in how much they were enriched in fat. So at that time, it wasn't straightforward that you had more fat and you had more liver disease. So we genotyped the SNP in a cohort of people with liver biopsy. And we could see that, in fact, it was not only fat, but also inflammation and fibrosis. Then we thought about cardiovascular disease. And they said, let's look at what if they have more atherosclerosis in the carotid artery. And we look at that. There was ultrasound in a subgroup of people. And in fact, we saw that, contrary to our expectation, they had lower carotid plaque, so less atherosclerosis. We thought that that was somehow a mistake or just maybe a finding by chance, and we sought to replicate the data in the Swedish obese subject study. This is a large cohort of 2,000 obese individuals followed for 15 years. We look at those people and genotype, and we could see that it was true. People with the variant had lower myocardial infarction. So this variant dissociates all this talking about cardiovascular disease and MASLD, indicating that maybe it's not true, maybe it's only epidemiology. However, we also look at lipoprotein levels because the most important predictor or risk factor for cardiovascular disease is dyslipidemia. And so we look at liver circulating triglycerides, and they were lower. This is compatible with the model in mice. However, what it was also lower was also ApoB. And if it had been like the mouse model, you would have found lower triglycerides, but not lower ApoB, because the number of particles wouldn't change, so there wouldn't be any difference in the two. So we wanted to understand more. We thought there must be something with ApoB there. So we look at spheroids. Generated them with primary human hepatocytes. When you culture them together in a round shape, well, you see that in a week they form a 3D sphere structure. And then we challenge the spheroid with fatty acid exposure, incubated with fatty acid, with 2 different doses. And then we look at the amount of fat in the spheroid. And you see here the blue are the nuclei, and the orange is the amount of liver fat in the spheroid. And you see that it doesn't matter if you don't have or you have low fatty acid concentration or high, the mutant has higher liver triglycerides, indicating that the model recapitulates the primary human phenotype. So then we look at ApoB levels. And what we saw was that in the media, there was much less ApoB as compared to the wild type. We had 3 wild type donors and 2 mutant donors, and there were similar results in all 5 of them. These are human donors, so it's not easy to find many of them. So then we thought, let's look within the cell, in the cell lysate, and we saw that it was also less in the cell lysate, so less apolipoprotein B. So if the lipoprotein B had been stuck in the cell, you would have seen less out, but more in. However, it was less in also, indicating somehow there could be some effect of TM6SF2 in protein stability, in ApoB stability. We ended up the study there, postulating this model. And a couple of years later, this is an independent, I don't know, these guys from Korea. What they could show was that indeed TM6SF2 stabilizes apolipoprotein B with 2 intraluminal loops, indicating that the absence of TM6SF2 would cause degradation of the protein. And so if you put all this together, what about liver disease and cardiovascular? The model is that carriers of the variant have higher liver fat because it gets stuck in the liver, less lipoprotein, that's why it gets stuck, and therefore also less cardiovascular disease, because this is the main predictor of that. This model is a paradigm of liver disease. Just please keep that in mind. I will remind you later. Let me tell you a little bit about MARC1 now. So MARC1 is a protein that sits on the mitochondria, on the outer membrane of the mitochondria. And a genome-wide association study, this is an American group. We also did it almost simultaneously. What we could, and they could show, we could show was that there is a signal with the MARC1 variant on transaminases, ALT, and carriers of the variant have lower ALT, indicating that this was a protective variant. So the variant was inducing an amino acidic change from an alanine to threonine in 165 position. So we wanted to understand more. So this is a protective variant. How is this working? Loss or gain of function? We thought also this could be used as a target. So we cloned the MARC1 protein with the wild type variant and with the mutant variant in a vector, and we overexpressed it. The protein was tagged with a V5, so you see the beautiful blot here because it's a V5 tag antibody. And what you can see is that there is less of the mutant protein here. So the 165T is less than that. Was about 50%. But then, of course, we thought, but maybe there is some problem with the overexpression vector, so we measured mRNA, and mRNA was higher indicating maybe, I don't know if it could be a compensatory mechanism, but anyway, that the reduction we see here was not due to lower expression, mRNA expression, but something that was related either to synthesis of the protein or more degradation. And the data was also similar. This is just a different cell type, HUH7. These are FG2. The vectors are the same, but we thought to repeat it in a different cell type to make sure this was true. And we also looked into human donor here. These are primary human hepatocytes from carriers of the wild type and the mutant. And you see that there is always a lower protein level in those with the mutant. And it's about the same quantity of reduction. The magnitude is the same. And in this paper, if you're interested, we also figure the mechanism. So what happens with the mutant protein is that the protein gets ubiquitinated, and it undergoes proteasomal degradation. So the mechanism by why you see less protein is due to proteasomal degradation. So this data showed that this is a loss of function protein. I mean, the proteasomal degradation, and there's less protein, so loss of function protein. So we tested whether if we would knock it down in cells, you would get a better, lower liver fat content. And these are the experiments in primary cells. And you see that this is the wild type protein, so the functional one, and this is downregulated with a specific anti-MARC1 siRNA, and these are two different fatty acid concentration, 330 and 300. This is quantification here. So it's a robust phenotype. Doesn't matter if you challenge it or not. There is a reduction in the amount of fat. This is the red part here that you want to look at, the all-red-all staining. So then we run tracers to figure what is the mechanism behind. Is this lower de novo lipogenesis? Is this more beta oxidation? Or maybe less mobilization from lipid droplets? And the study, this is only the positive result, and you can also look at all the other pathway. So what we could see was that when you would knock down the MARC1 protein, you have more beta oxidation in the cell. So this is the way it changes the amount of fat in the hepatocyte. And there's also 2-3 companies with MARC1, SI, ASO, RNA trying to targeting fatty liver using this mechanism. And this is interesting because the first drug approved for MASH, I think it's been alluded before, is a thyroid hormone agonist that increases beta oxidation and specifically in the hepatocytes and reduces liver fat. So indicating this could be a good strategy to go forward with. So taking all this together, it seems like MARC1 is sort of like a gatekeeper of fatty acids in the mitochondria, and if you remove it, somehow maybe there's more possibility of fatty acid to enter mitochondria to be oxidized. So GWAS is what I started with, and I still like them also. A couple of years ago, we did a genome-wide association study in the UK Biobank, 40,000 people. What we could find was with multiple adiposity adjustment. So we found a bunch of novel loci here. So together with the old one, we were on 250 loci altogether. We replicated some, so we ended up with 30. But what we thought about doing was something a bit different. So we tried to dissect the heterogeneity of the disease by generating 2 polygenic risk scores. Based on what? Based on this lipoprotein retention paradigm that I talked to you about. So we generated 2 polygenic risk scores. One that was mimicking lipoprotein retention, with SNPs mimicking that, and the other one with SNPs that do not mimic that, and then supposedly they would change either lipogenesis in the liver or beta oxidation. as opposed to secretion with lipoproteins. And then we look at the outcome. So this conundrum of the systemic disease. And we started with the lipoprotein retention that in fact was our positive control because we knew what would happen. So what would happen is liver disease, we knew it already, protection against cardiovascular disease, and this is because, like I showed you, there is less lipoprotein particles, a little bit of risk of diabetes, and that's it. The surprise came when we look at the other polygenic risk score. And here you saw a bit of less liver disease, but the entire association with all the diseases that I showed you, heart failure, much larger risk of diabetes, much larger, hypertension, chronic kidney failure, cardiovascular disease. This is the cross-sectional analysis. We also did the prospective analysis in the UK Biobank. And if you look at the dots here with the events, it's very much similar, indicating that there may be at least two types of MASLD. One that is, due to this lipoprotein retention, that is mostly liver disease, and then this other that is embedded in the entire systemic disease with kidney failure and kidney damage and heart, coronary artery disease, diabetes, hypertension, and everything. So about a year before, different projects started. This started with completely different premises. It was a completely clinical project, nothing genetic related. And the idea was to find a way to making clusters of people with MASLD, starting from very simple clinical characteristics that, biochemistry that you find, or clinical like BMI, age, LDL cholesterol, ALT, HbA1c, and triglycerides. So the analysis was done in a cohort of bariatric surgery people, almost 1400s, in which there was liver biopsy, and it was used an unsupervised clustering model. This is called k-means clustering. And so when you fed all these parameters, the software came up with 3 different clusters. A red one that was characterized by having higher ALT, a little bit higher LDL cholesterol, and one cluster that was characterized by having higher HbA1c, so diabetes, blood glucose, and higher circulating triglycerides. And then there was a control cluster. So first was to look at liver. So what happens in the 3 clusters here? What happened was that there were these 2 clusters with the same amount of liver disease, steatosis or inflammation or fibrosis was the same as compared to the control. 2 clusters, different phenotypes, same liver disease. And of course, we wanted to replicate the data in an independent cohort. So we went on to another bariatric surgery cohort, and you still see the 2 clusters, same disease. And also in the UK Biobank, in people at risk for MASLD. And you see again that the cluster stood out similarly, same liver disease, same liver disease. So the question was, okay, we have different clinical phenotypes, if you want, same liver disease. Do they have also different or similar clinical trajectories? So first we look at the liver disease, and maybe not surprisingly, this is the red. I will point the red to you. So the 2 clusters were similar in liver disease. I think this makes sense because they had also similar liver disease in histology. However, what was different was cardiovascular disease. It was basically only in this that then we call cardiometabolic because of the HbA1c and triglycerides and the outcome. While in the one that we call liver specific, there was no cardiovascular disease whatsoever. So, and also diabetes was not the same because the cardiometabolic had a large excess of diabetes onset, while the liver specific was there, but it was much less. And these data were very similar to the genetic data, although they were not meant to be going to the same endpoint. That's how it happened to be. And so, yeah, we were lucky enough to published them back to back into Nature Medicine. And this is interesting for clinicians because, again, starting from simple things that everyone basically would have in a biochemistry profile at the clinic, you can really see, okay, this person will have a cardiometabolic disease. Here you would have another person with only liver disease and then the control area. So basically, proof of concept that, in the future or now we can have a better grasp on whether you will have only liver disease. This is at a clinical level, while in those that, you would want to target also more aggressively lipoproteins for cardiovascular disease, for example. And the genetic data provided the concept that, the liver disease, lipoprotein retention, protection, cardiovascular disease, a little bit of diabetes. And then you had this other MASLD, we call it systemic, in which you also had the liver, hypertension, diabetes, and maybe all driven by visceral adiposity. This is more of a question. We didn't look at that. So it was mostly this part. So putting them together, we could find these two different types of MASLD. And you may think, oh, now maybe we can do genetics with the polygenic risk score and we'll figure in the clinic. No, you can't do, because these are very small effect size. So it's only about proof of concept, trying to tease in causality, because genetic variants are randomized at birth. So whenever you have an association with human genetics, Steve will tell more about it, he knows much more than me, you can get a better sense of causality and what's happening. I need to finish. Yeah. Well, yes. I have the last part that I want to show very fast because I want to show some last data on ApoB that are just published. So, and I'll go straight to the point. So we look at mutations in ApoB in people with MASLD. It's much more there. So 1 in 20 have mutations in this ApoB gene, so this lipoprotein retention. And why I'm telling you this now? Because yes, it increases cirrhosis, it increases cancer. This is cancer. But what is really important here is that it segregates in families. So this is the first variant that we can really see, okay, proband with fibrosis cirrhosis, son with steatosis fibrosis. And this is the first time that it's formally tested. We can say there is a positive transmission test indicating that, in the future, I hope this gene will be screened in the liver clinic to find people in the families, and you can prevent those in the families. So this is just published in JCI. I just want to show you a little bit of more really new data. Thank you to Luca Valenti, all the collaborators, my group, and the funding agency. Thank you.
Thank you so much for a very interesting talk. Questions from the audience? Yes, Steve.
So, congratulations on beautiful work you've done since you managed to escape from Cambridge all those years ago. You've done
it was a good learning experience also there, so it really helped.
You've done a fantastic job. So mine is more a comment than a question.
Yeah.
And it relates to, you finally got to the word adipose tissue after 45 minutes. And in fact, when I sit in the lipodystrophy clinic, every patient I have with lipodystrophy has fatty liver disease.
Yes.
So I think almost certainly the cardiometabolic drive, the 50% segment that isn't primarily driven by liver, is probably very likely to be driven by adipose tissue dysfunction. Not necessarily visceral fat. I think visceral fat may be a passive bystander to some extent. So I'm just going to say that I hope my talk at the last day will complement your talk by suggesting that a lot of that other liver pathology is likely to be driven by a primary disorder of adipose tissue, which is driving the lipid into the liver and not intrinsic to the liver, but is essentially overloading it because you can't store the fat in say. And I just think what would be useful would be to look at the genes in your GWAS as the GWAS data gets larger and larger for LFTs, to try and parse out which of the genes are specifically enriched in adipose. I mean, PPAR gamma was sitting up there as one of the obvious ones. You didn't even mention it. It was almost as high as the others, but wasn't mentioned. So I think adipose tissue is likely playing a big driving role in the other 50%.
Stay tuned. That's my answer. This is not, no, let me tell you. So there is a hopefully soon Nature Communication paper with single nuclei showing that when you have steatosis, what happens in the visceral adipose, it doesn't differentiate. But in the subcutaneous, still you have differentiation. So this hypothesis that first you don't expand the subcutaneous and then it goes in the visceral may not be true. So the data suggests that it in fact is the visceral. When you can't expand that, it goes in the liver. So it's sort of like fine, get out of the window, going to the liver. So those are coming, and I agree. Also genetically, I'm working on that. So I hope to come something putting also together pancreatic fat and a bit more holistic. Yes, thank you. You see, I learned something.
So are there more questions? Yes, there's a question over here.
Hi, really interesting talk. I'm really puzzled to see the fact that you have this liver-specific phenotype, but then there's also this systemic different disease, even though they present as similar phenotypes. How do you think we can even begin to really unravel these mechanisms? And again, going back to that causality, right? Because of course, all these organs are interconnected. We are all the time communicating with different organs, and how could you have only liver phenotype, whereas the other one develops in the cardiovascular disease? You have, of course, shown that why that would be protective effect with the lipid droplets kind of staying there. But how do we even begin to kind of tackle this? And of course, how does this go into future therapies as well?
Right. So I think a bit the previous question was, on the similar way. So we need to look at other tissues also. The visceral one is not, I'm not convinced yet that it's the liver primarily causing everything. It may be mediated by visceral adipose or pancreatic fat. Another question is that when you do this polygenic risk score, you have only one mechanism, lipoprotein retentions, versus the other. So, but then you need more variants to do the next step onto more polygenic risk scores to look at what happens if you increase like beta oxidation or you reduce that. So this is kind of what it's coming next in exploration of the data. And for therapeutics, so the data show that if you lower fat, you get a benefit for the liver. So that's like, something established. The question would be, is there a difference if you reduced de novo lipogenesis or beta oxidation in the other organs? So this I don't know. It may be, it may not be. For the liver disease, I don't think it changes whether you reduce one or the other.
There's a question up there.
Yeah, Charles Brenner, City of Hope. I was really impressed with the work that you did on the common variants like PNP and some of the others.
Yeah.
But I'm questioning whether we classify these as loss of function or gain of function. So in particular, when you can knock down the effect of the mutation, with an antisense, don't you have to concede that it actually has a component of gain of function? And then secondly, with how frequently some of these exist in the population, do you think that they were enriched to get us through famine, throughout human evolution? Right.
So what do we do? So first of all, this seems to be a redundant system. So it's not that if you don't have ATGL, it's really bad for you. It's like people with ATGL mutations are doing really bad. There's no evidence if you remove PNPLA3 that you have any actual phenotype. Because I think it's redundant. And so if you don't have that, something or something else will do it. For the gain of function, I'm pretty sure there's a loss of function for this remodeling of lipid droplets. This gain of function, I can understand if you think of the CGI58. I kind of failed to grasp how it can be a loss of gain of function at the same time. But sometimes we don't, things are just like the way you are, like they are, like GIP before GLP-1. Some things are not really, reconcilable based on what we know. So I think that the data show that if you remove it is good. Of course, for the long-term data, we just need long-term data. There is no genetic signal that it's bad if you remove it. And I believe that there's some kind of selection pressure due to either famine or the other force for shaping the genome was diseases, infectious diseases. It could also be infectious disease. But then, of course, the famine problem, at least now, it's not an issue anymore. So if it was, protecting against that, it's something that it's like, even if it's gone now, you wouldn't see any bad effect. And because you would treat fatty liver that comes with overnutrition, if at some point there were going to be no food in the world, you're not going to need to target the protein anyways, because there will be no fatty liver. So,
a final quick question up there.
Over here. My question is in connection again to also the targets. When you were showing PNPLA3 and the apolipoproteins B and E, they're very pleiotropic, right? So my question is, how would you go around this? Because it's not a surprise to me that you see a differential cardiovascular risk if these variants are reported for chronic cardiovascular, kidney, and metabolic diseases. So my question is, how would you go around this target approach if you have to do it again? And have you considered focusing on liver-specific variants that you're clearly sure that they're liver-specific, and then see whether they have this cardiovascular risk differential effect?
So I didn't show the data, but we look at EQTL in liver biopsies, and it turned out that the variants in this liver-specific are liver-specific. So PNPLA3, it's basically only expressed in the liver, a little bit in the retina. So it's a liver-specific protein. TM6SF2 is a liver-specific protein, a little bit in the intestine, because also there you do chylomicrons and ApoB48. So these targets are, MARC1 is also liver-specific, so these targets are liver-specific. So you wouldn't need to think of systemic effect. But of course, if you would, restore TM6SF2, you would increase lipoproteins in the circulation. So you can't really do that, because you would make liver better, but increase the heart. Of course, you could target the lipoproteins with a statin or PCSK9. But there is another example I didn't talk about, is HSD17B13. That's also liver specific. So all the targets that have been developed for liver disease are indeed proteins that are present exclusively in the liver. For the other genetic variant, I think too much of a small effect size to start thinking targeting, at least for now. I hope I answer the question. If not, we can talk in the break.
Let's give Stefano
thank you.
another hand.
Thank you. Thank you.
Thank you so much.